Hi there, thanks for tuning in. Welcome to my presentation, LSV-Seq, a novel targeted sequencing method to measure alternative splicing. My name is Kevin Yang, and I'm a MD PhD student from the University of Pennsylvania with the Barash and Choi labs. To get started, I first wanted to review how we generally study splicing using standard short-read RNA sequencing, which involves capturing transcripts in an unbiased manner using oligo-DT primers, resulting in the following distribution of reads. Most of the reads are exon body reads that don't span junctions and are less important for splicing measurements. Only a small fraction of reads that we get back are the junction spanning reads that are truly critical for quantifying splicing. We can quantify alternative splicing using junction reads as a percent spliced in or psi value. For instance, the psi value of the green junction here would be the fraction of green reads out of the total reads, which in this case is the green plus the tan reads. To summarize, this diagram highlights how the major fraction of RNA-seq reads do not in fact span junctions. If we could come up with a method to somehow capture only the most informative junction reads, this would drastically improve coverage of splicing events we are specifically interested in. This would both reduce costs, allowing us to sequence more samples more deeply, as well as increase sensitivity, allowing us to detect splicing into contexts that we previously couldn't. The algorithm our lab likes to use to study splicing is the MAGIQ algorithm, which thinks of splicing in terms of what are called local splicing variations, or LSVs. For instance, MAGIQ would divide this cassette exon splicing event I've shown here on the right into a source LSV consisting of all of the junctions coming out of the 5' end, and a target LSV consisting of all of the junctions coming into the 3' end. As an aside, I encourage you to check out our lab's poster number 1203 about the new MAGIQ V2 algorithm with Paul Jewell to learn more. I was initially interested in using MAGIQ to quantify splicing in the GTEx heart left ventricle dataset, which has almost 200 samples. However, I realized that a significant number of events in this dataset are poorly covered. Here I've binned all the LSVs we get back from MAGIQ into different bins of coverage on the x-axis, while the y-axis plots either the number of LSVs per bin in orange, or the fraction of samples in which we were able to quantify given LSV in blue. You can see that in lower read coverage bins, we are only able to quantify the LSV in half or less of samples. To put this into context, bins with less than 20 reads constitute around 30% of all detected LSVs, which is a very large fraction. So given this, I wondered how I could systematically and accurately reassess these events that we are losing coverage on. Our new targeted method LSV-Seq is inspired by a previous method called MPE-Seq. Specifically, we are proposing to enrich for splicing events by creating a pool of thousands of region-specific primers so that we're only capturing splicing events that we actually want to. As shown in this diagram, this leads the junction spanning reads rather than the exon body reads to now make up a majority of the sequence reads we get back. Finally, LSV-Seq derives its name from how it is directly tied into the concept of LSVs from the MAGIQ algorithm. Specifically, we are aiming to pick up target LSVs consisting of 3' splice site adjacent junctions. Early experiments, however, showed that targeting the human transcriptome using LSV-Seq was challenging. When we tried to use the MPE-Seq method optimizing yeast directly in human cells instead, we found we only recovered about 1% of reads targeting events we actually wanted to capture. This makes sense since yeast have much fewer splicing events in general and lower transcriptomic complexity compared to humans. So to successfully target splicing in the human transcriptome, we needed to devise a much more stringent procedure which can overcome the significantly lower signal-to-noise ratio for each target. To address this, we came up with a new workflow for LSV-Seq optimized for human cells. First, we select thousands of events of interest from a more shallow exploratory dataset generated using standard RNA sequencing. Then, for each target region of interest, we generate all possible candidate primers and select a single primer predicted to perform the best. In order to optimize prediction of high-performance primers, we built a model to learn trends related to primer performance, hoping to capture, for instance, the strong positional nucleotide trends we observe consistently, such as how 3' adenosine-rich primers perform consistently better. I've created a training dataset consisting of 2,000 data points across 3 previously sequenced LSV-Seq libraries. The features we fed into our model include predicted on and off target binding scores based on based on transcriptomic alignments, primary and target characteristics like melting temperature or length, as well as encoded nucleotide positional identities. The target variable we are predicting is a metric we are terming the fraction primer binding, or FPB. To calculate the FPB, we use BLAST to first assign all of our read alignments to their primers of origin, then calculated what fraction of the primer was actually bound to the transcribed RNA, which here consists of the fully bound portion of the primer in dark blue divided by the total length of the primer, including both the unbound region in orange as well as the bound region in blue. Based on our training data set, we were able to use a boosted decision tree model to achieve a cross-validation Pearson's correlation of 0.8. We also observed that when we looked at the top 15 features in our model, we see that many are expected, including the strong presence of adenosine-specific features. After predicting the primers we think will perform the best, we order the primers in a single pool and use them to create libraries using our optimized preparation protocol. We use the pool of primers in the first reverse transcription step, in which we use highly stringent chemical and temperature conditions to ensure selective binding of the primers to the target RNA. Of note, in addition to a unique targeting region, each primer has a UMI which allows us to deduplicate reads after sequencing. To avoid significant exponential bias from PCR, we then linearly amplify the resulting cDNA using an in vitro transcription procedure, then finally add the second library adapter, perform a low number of cycles of PCR, and sequence the final library. Finally, after sequencing, we used a custom version of Magic to analyze the targeted splicing events. The current best version of the protocol was demonstrated in a pool of about 400 primers in jerkcat T cells. Here, we observed the relative fold enrichment of each targeted event in LSV-Seq compared to RNA-Seq, with a median of over 500-fold overall. Another metric we looked at is the percent of the total library mapping to target events, which is about 70% for LSV-Seq, while it is just a fraction of a percent for RNA-Seq. Furthermore, we were able to cover events that have particularly low coverage in standard RNA-Seq, such as lowly expressed events. Here, we separated our fold enrichment graph from earlier into different bins of expression, with bins on the left having lower expression and bins on the right having higher expression. We see that for the lowest expression bin, we are able to capture a fold enrichment of around 700-fold, which shows that we are able to cover those events that are typically poorly covered by RNA-Seq. Having established that we greatly enrich for the targeted events, we next looked at quantification accuracy of LSV-Seq relative to RNA-Seq. Here, each LSV that we targeted is reduced to a single junction, which is selected at random to be either the maximum value or the minimum value junction. We see that we achieve a reasonably strong correlation overall with RNA-Seq of 0.88. As a caveat, while the LSV-Seq data shown here was generated by myself, the RNA-Seq on the y-axis is a previously published data set from our collaborators, not generated by me. This means that discordant points could potentially be due to biological sample-based differences rather than technical differences. We also looked at the coverage of individual LSVs. Here we are looking at the cumulative distribution graph for the number of reads of coverage in the RNA-Seq dataset in orange, with about 130 million total reads of coverage compared to LSV-Seq in blue, with about 10 million reads. In contrast to the much worse coverage in RNA-Seq, we see that in LSV-Seq, over 90% of the events have at least 100 reads, which represents exceptional levels of coverage. And in perhaps the most visually striking example of the improvement we achieved with LSV-Seq, we last looked to see if we detected previously unidentified junctions. Here in this graph, we are plotting the number of extra junctions observed in LSV-Seq compared to RNA-Seq. A positive number represents more junctions in LSV-Seq, while a negative number represents more junctions in RNA-Seq. We see that the mode of the distribution is at 0, which makes sense since in these cases LSV-Seq is perfectly concordant with RNA-Seq. However, we also see a large right tail, which represents cases in which LSV-Seq is detecting many more junctions compared to RNA-Seq. So we wondered what was going on at the extreme tail end. To pick this apart, we used our Voila visualizer tool. Here, the same splicing event is shown on the left in RNA-Seq, and then in LSV-Seq on the right. We see just 3 junctions with low coverage in RNA-Seq, while we see 21 coverages with the greatly improved coverage of LSV-Seq. The same three junctions on the left in RNA-seq represent the major junctions with now thousands or hundreds of reads of coverage on the right in LSV-Seq, which are also boxed in their corresponding colors. We think there are many different potential mechanisms by which the de novo splice junctions we see here that we are still trying to validate arise. These phenomena might include noisy or transient splicing, noisy transcriptional start sites, or technical artifacts. However, we think it's less likely these junctions are simply artifacts, since 20 out of 21 of the junctions that we detected can be found in a database of all splice junctions across GTEx or SRA. In conclusion, we use LSV-Seq to target thousands of splicing events of interest using a new computational and experimental workflow that's been highly optimized. We also demonstrated that LSV-Seq can greatly enrich for the splicing events that we're targeting compared to standard RNA-seq, as evidenced by both the greatly increased coverage and fold enrichment, as well as the improved detection of de novo junctions. For our future directions, we hope to first validate LSV-Seq as a technology by seeing how well we are able to capture and quantify the poorly covered splicing variations in GTEx that I showed you in the intro slides. Once this is established, we're especially interested in trying to capture transient or rare splicing intermediates, for instance, those that occur in cryptic and recursive splicing. We're also interested in specific contexts with these types of splicing intermediates, like cancer therapy resistance or the cell-free transcriptome. Finally, once the bulk method is well established, we'd like to develop LSV-Seq into a single-cell protocol to accurately quantify splicing robustly in single cells. And last but not least, I'd like to acknowledge both Peter's lab at the Children's Hospital of Philadelphia, as well as Joseph's lab at the University of Pennsylvania, for supporting me throughout this project and my PhD so far. I'd particularly like to shout out Joseph Eicher and Paul Joel from Joseph's lab for their help in adapting Magic so that I could use it to analyze LSV-Seq data. Thanks for listening, and please reach out if you have any questions at all. My email is on this slide, or you can come find me in person at my poster, which is number 992. Thanks.